if someone gives a gene needle in the time, overall we try to work out to try to mark down. Yeah, including fitting. But don't worry, if you have more time.
Okay.
a little bit more.
Okay.
Good afternoon, everyone. I hope everyone enjoys summer as well as the lunch, which is always good. So today we have another faculty candidate, Dr. Zhang, who is applying for a research assistant professor in my lab. Also, it's a faculty position in the school. Dr. Zhang received a PhD in Harbin Institute of Technology in China, and then he has done a few postdoc trainings, including the most recent one at University of California, San Diego, and Indiana University at Indianapolis, IUPUI. Dr. Zhang has done for many years the bioinformatics research and also support. Currently, he is he holds a faculty position, research assistant professor in bioinformatics, and his primary bioinformatics people in the University of North Texas Health Science Center near the Dallas. It's not far away, but still you need to take a flight here. And also, Dr. Zhang has done, again, as I mentioned, not only bioinformatics, but he also has been interested and has done some imaging analysis, which potentially can be very helpful in nowadays, big data, especially trying to understand the precision medicine, because imaging data is very helpful for clinical relevant information. So I will give you more time. So yeah, talk, the title is here, and hope everything's fine. Okay.
Yeah.
Good.
Thank you. Good afternoon. My name is Sam. I'm from University of North Texas at Health Science Center at Fort Worth. My primary job is to provide the bioinformatics service for the faculty and the research fellows, so such as data warehouse, data storage, and data analysis, and develop some web tools and web application, and also establish a research collaboration with experimental scientists from different departments. Yeah. I also did some my own project, and protein isoform identification is one of them, as well as like pathway analysis and biomarker identification. So today I'm gonna talk about them. The clinical proteomics is the application of proteomics techniques used for the medicines, and they try to solve a specific clinical problem in the context of clinical studies. And it provides us with opportunity in more effective strategy for early detection, disease detection, and monitoring. Also, more effective therapies and developing a better understanding of disease pathogenesis. Protein isoform is a form of different proteins, different forms of the same proteins, and with slightly different amino acid sequence that have the diverse activity and functions. So normally when we say a protein isoform, we refer to three types. So the first type is alternative splicing. So alternative splicing is it occurred during the splicing process, in which intron removed and exon remained, combined, joined. And the second type is the single nucleotide polymorphism. And the third type is the post-translational modification. So post-translational modification account for 90% of the protein isoforms, and the mRNA variants account for like nearly 8 or 10% of the protein isoforms. So as a new classic diagnostic biomarker, protein isoform has been reported in many cancer cases.
So the people in the backside can you hear clearly? Otherwise, maybe you can speak more. It's okay for you? Okay,
Okay, good. So here shows the this is the sleeves, and here we can see the red one, red axon is skipped. So the yellow one and the green one combined together. So this is the axon skipping alternative splicing. The post-translational modification as a protein isoform can be identified using some search engine like Amazon or Sequest, and Moscot. So my research focused on identification of the alternative splicing, but currently existing the alternative splicing database for the identification in the proteomics has not enough coverage. So I first developed a database which is the synthetic alternative splicing database for this purpose, trying identifying the novel isoform. The whole flow chart contains 3 steps. So the first step is extract the gene sequence and gene structure from the ensemble database and build the synthetic alternative splicing database. The second step is run the search engine Amazon against the synthetic alternative splicing database with the raw tandem mass spectrometry raw data. And then we do the preliminary data analysis and find out the alternative splicing. The last step is for the validation. So we have 3 strategies for the validation. The first one is the literature review, so we can see this alternative splicing biomarker has been reported or not. The second method is the cross studies, cross validation. We use the different samples from different labs to validate the same alternative splicing biomarker. And the third one is to biological interpreting, such as pathway analysis, try to interpret the biological meaning for those alternative protein isoform biomarkers. So that's the 3 strategies. So this is the web interface for the SASD database. So here is for users to input the gene name and the protein name. So users can input different type of genes or proteins. For example, you can input gene ID or gene name or even unigenes. And for the protein, you can input a UniProt ID or UniProt access number or IPI IDs. You can even just copy the text from web page or from PDF file or DOC file, just copy here, no matter what kind of format it is, or no matter what's the delimited character. Like you can use the comma, you can use semicolon or lines or even space. This text box can automatically recognized genes or proteins. So it's very convenient for you to upload the gene list or protein list. So often you upload the data, the gene list, you click search, and the figure B here is the result. And it shows the relationship between the moleculars and the genes and the transcripts and the relations, like types, what type of the alternative splicing, and what's the model, and also it contains some enrichment score like p-value and relative expression value and the number of proteins in that pathways.
So how the enrichment score was calculated?
There's multiple methods. One method is use the chi-square testing method. That's
basically my question. For what purpose? How can you measure the enrichment?
The enrichment is calculated using the chi-square testing method.
My question, like enrichment is more like a hypothesis. You test A is statistically enriched with B. So what is this kind of, for what purpose?
The enrichment is for the gene list you input against the whole genome.
Against whole genome?
Yeah. That means how many possibilities you can see those gene lists can occur at the same time for this disease. Yeah. When you compare to the whole genome. Yeah. And also I have other method to calculate the enrichment score. Some method is like very simple. How many the genes show in that pathway. So normally if a pathway contains like 100 genes, but you identify like 50 of them present in your gene list, in your inputted gene list. That means half of them. That's very, very high, enrichment score. And also, I use another measurement like the relative expression value. Relative means, like just now I said, if we have 100 genes, but we find out 50 of them, so that's 1/2. But how about we have only 10 of them, 10 genes in that pathway, but we identified 5. We identified 5 genes listed in that pathway. So which is higher? So I use a measurement called the relativity. So for the relatively gene expression value, the 50 over 100 is higher than 5 over 10. Yeah. So the figure C and D is for the gene view and the transcript view. So for the gene view, here show all the alternative splicing for that single gene. So this is much larger than the transcript view. Transcript view contains all the alternative splicing in that mRNA. And also show the alternative splicing view here. We can see the different part. For this one is a single exon, so all shows just in one color. If we have like 2 parts, like exon, intron, they will show in different color here. And the last figure is show the association with the pathway and the disease and the drugs. So this is for another database I will introduce later. That's called integrated pathway analysis database. So show here is TP53 is associated with apoptosis pathways. Yeah, this one is linked with the P53 protein. There are totally 6 types of alternative splicing events we considered now. The first one and the second one are from the normal peptide. So by normal, I mean no any alternative splicing involved. So this one from the single exon, that means the peptide from the red bar only. And the second one is from the labeling exon combined. So by such as like exon 1 directly combined with exon 2, or exon 3 combined directly with exon 4. The third one is non-labeling. Non-labeling means some of the exon in the middle of the junction will be skipped. So this is also called exon skipping events. For example, exon 1 combined with exon 3, so exon 2 is skipped. The fourth and the fifth are for the intron action, intron axon and axon intron combination. So this one is for the right intron retention, and this one for the left intron retention. The last part is for the peptide, just coming from single intron. So this is coming from the single intron. All the three parts, axon intron, intron axon, and a single intron, are called intron retention effect. Yeah. So there are totally 6 types of alternative splicing we consider in current database. So this is the example. We do the identification or analysis from the plasma breast cancer proteomics. The data we have contains 80 samples. So 40 of them are the women diagnosed with breast cancer, and 40, the rest of 40 is the healthy women used as a control. For example, the first one is from the intron 8. Yeah, so this means it's the single intron alternative splicing. It has the p-value 9.4 power -5, and it present like 20 of them present in the cancer samples, and 4 of them present in the health samples. So we didn't find it when search at the Peptide Atlas website. So probably means this one is the larval sequence, peptide sequence, and is from the famous gene, probably breast cancer genes, ERBB2. The second one, the red part is from the exon 7. The green part is from the exon 11. So the middle exons, like exon 8 to exon 10, are all skipped. So this is the exon skipping alternative splicing. So currently the top hat can use to identify the alternative splicing from the RNA sequencing in the mRNA level, but top hat cannot handle, so long separated the exon combination. Top hat can handle like exon 3 combined with exon 5, or maybe exon 4, but cannot skipping like 4 exons in the middle. So this is the power of using some synthetic exon combination method to build a man-made database and then used for the alternative splicing identification. Either you use it for the RNA sequencing in the mRNA level, or for the proteomics for in the protein level. Yeah. But the method is quite useful. Then
here, maybe ask you a very short question. Can you tell us which kind of data you used here to generate these results?
This is the result is from the protein plasma data.
Protein plasma.
The proteomics. Is now probably is stored in the CPTAC website, but CPTAC doesn't contain the raw data. You can have the result, but that data cannot be used for this purpose. It can only be used for identify some of the normal markers. So in order to get this, you need to have the raw data. The raw data not published yet. Yeah.
So this is CPTAC or not CPTAC?
This is from the CPTAC. This data is from CPTAC. Yeah. Part of the CPTAC data, especially for the processed the CVTAC data already publicly available.
Yeah, we published just published ovarian cancer CVTAC data in Cell paper.
Oh.
Yeah, and a year ago they published colon cancer.
Yeah, they have quite a. At that time when I was in Indiana University, the data we have is breast cancer only. But now they have like ovarian cancer and or even has the lung cancer now. Yeah.
Wait, so what's the comparison here? Is this comparing
health with the cancer?
People that don't have cancer versus people that
Yeah, like this 20 cancer samples versus the 4 the health sample.
Okay. And are you saying that the healthy samples have HER2 but not the splice isoform, or?
What I say is the 4 health sample have this peptide sequence.
Okay.
But we have 40 health samples, so only 4 of them have identified this peptide. That means 36 we didn't find it. We didn't find this peptide.
Right. And then out of the, but 20 of the breast cancer patients have this peptide?
Yeah, that and 20 not know this peptide.
And out of these 20, how many of those patients have HER2 amplifications?
Sorry?
How many of those patients have overexpression of HER2? I mean, I'm just wondering if...
I see. 20. Yeah. Because I used some kind of threshold to convert that expression value to like 1 and 0. So here just shows if 20, that means the 1. The 1 or 0. 1 means present, 0 means absent. So that means the 20 samples of cancer has this the first peptide.
What's the meaning about the p-value?
P-value means the probability for the differential expression between these 2 groups. Yeah. Similar to the traditional gene expression data analysis. So here I will show another example. Yeah, the third one. Third one is the intron retention. So the red part from the intron 2 and the green part is from exon 3. So this is the intron, left intron retention effect. Yeah. So all the 8 biomarkers has a p-value smaller than 0.05. Yeah. And they are not identified, not mapped with the peptide atoms. So probably they are the novel one. So in order to confirm this, we do the UCSC genome browser searching, and the result further confirm this. There's no hit from the genome browser. For example, the first one, there's no sequence from the reference sequence for the first peptide sequence, like this one, first sequence. And the same for the rest 7 peptides. So that's the 2 methods we use to identify the sequence is new or not. Next, I will introduce the pathway analysis database. The idea for this database is traditional pathway analysis, like the famous one, ingenuity pathway analysis, IPA, can only be used to identify the association between like genes, disease, gene, organ. Gene organ means tissue specificity, and gene pathway or gene drugs. So show here is the black line. Yeah, show here. And we call this observed association. But sometimes we want to find out the inter-association. So which is we want to know which pathway is linked with another pathway, and which disease is linked with which pathways. So that is the inter-association, which show here is the dotted line. So the iPad has the power to discover the both type of the association, even the observed association and or the inter-association, hidden association here. So in order to build this database, we extract the data from different sources. The first, we extract the genes and proteins from like Ensemble and NCBI or from the Uniprot, different website, or even from the IPI, different format. And the second data source is for the drug. We extract the information from the drug bank and pharma GKB. For the tissue specificity, is extracted from the database we created, called HOMA, for the tissue specificity. And for the disease, we extract information from pharma GKB and a comparative taxonomy genomics database. For the pathway, we extract the pathways from the following 4 sources. The first one is BioCarter, second one is CAG, third one is NCI, natural created, and last one is Rectome. Rectome contains much more pathways than CAG and BioCarter and NCI natural created. But the reliability in the Rectome is not as high as like NCI nature created or the CAG. The rest part is we calculate the similarity among those 5 components. We call it component, genes, disease, pathways, organ, and drugs, and do the enrichment analysis and build the inter-association. And then we use this method for the validation and see how many the level relationship we can find. So that.
So the pathway, what other pathway databases potentially you can use?
Well, the other pathways, as I mentioned, like IPA, is.
IPA you can download.
Yeah, you can download, but you can use it. For example, you upload your gene list and then see the enriched pathway, and then you can write down and you type it. But you cannot download it. Yeah.
How about the other publicly available pathway databases? More comprehensive. Are you aware of that? When did your iPad work with them? Yeah.
2012. This one was done.
If you do it now, what is your, which kind of update you like to do?
Now I think maybe there's like David. You can use David. David is downloadable, but in the tab format as a table.
Do you know pathway commons?
Yeah, pathway common and pathway common. Yeah, I heard of pathway common. So this is the interface similar to the SASD. So you can input your gene list or protein list at any format. And then the result shown in the figure B is for now. So C, D, E are for the different components. Here, B is for the pathway, the result for pathway, and C is the result for the disease. That means which disease associated with the gene list you input. And D is for the drug association, E is for the tissue specificity. And the G, H, I, J are for the inter-association between the pathways and pathway disease and pathway drugs and pathway the organs. That's the G, H, I, J. F is just for the gene ID and the protein ID mapping. And then next, I also did some the biomarker panel identification because some research paper says not the single genes make a contribution to disease. Probably is the disease is caused by a group of genes or proteins. So this idea is just try to find out, as a small number of genes or proteins as possible to predict or diagnose the disease. So we use this method for the peripheral blood breast cancer sample, which I downloaded from the GAO, and the number of biomarker is fixed as 5. So I use 5 because I read a paper which says the 5 or 6 number of genes or proteins as biomarker is enough for the breast cancer prediction. So there are 4 measurements I used, like sensitivity, specificity, precision, and accuracy. And the method I used just quite direct and straight. Just use the enumeration optimization just to find out. For example, we have 40 genes. We just randomly pick up the 5 out of the 40. So there's a lot of the computational times needed for this task, but the result is very accurate.
So let me ask you one more question.
Okay.
So those kind of formulas are pretty often used in machine learning, try to predict evaluate the outcome. Why you need all these 4 measurements?
Why I need all? Normally I just use one. I just use the AUC. I didn't put it just the area and the curve. Yeah. But why I use this? Sometimes if got the good AUC, but the specific is quite low, so probably we need rerun the program and change some of the algorithm. So that's the one of the reason. If the sensitive or specificity both are good, so we don't need to do the further analysis. For example, if we have very high like AUC is let's say like 75, okay, 75%, but we got sensitivity like 78%, but we have very low specificity like 50%. That probably we need to check the date, see the data. Maybe we didn't do the quality control. Maybe the normalization is wrong. So that's the reason why I need to use the 4 measurement instead of 1. But for the optimization, I cannot use the 4 measurement. I can only use 1. So I choose the AUC as the only measurement for the optimization method. Yeah. I know there's some other combination measurement, just a formula, just use these 4 and combine these 4 into the one formula, but I forget that name, maybe FOC or something like the formula. But using the AUC is reliable because most of the statistical paper, the reviewer just like the AUC result much more. For example, we identify here 4 panels. Yeah. So each panel contains only 5 markers. The first one, the top one panel, has the for the training set has 0.9053 AUC, and for the testing set is a little bit lower. It's 0.7879. Yeah. So you can see here. For the second one has the higher the AUC score for the training set, but lower the AUC score for the testing set.
Do you know roughly the sample size in each data set? That's the
yeah.
Sample size.
The training set, just now I mentioned I used the 80 samples. That is for the training set. And for the testing set, we have another 80 samples. Yeah. And also I have the third samples. But I not use it for
different studies or the same study?
Same study, yeah. They have study 2, study 3, and study 5. So this training study is study 2 containing 80 samples, and testing study is from study 3 containing 80 samples. And study.
If you do the breast cancer, do you need to consider the subphenotype of breast cancer, because breast cancer has the molecular profile,
right?
Subtypes very well defined. And
Yeah.
They are different.
Right, and depends on what purpose. If you try to find a subtype specific biomarker, you need pay much more attention to the subtyping. But you just find a general biomarker for identify the cancer versus the normal, so we don't need to consider that heterogeneity. Yeah. So for this purpose, no, we didn't consider subtype. But later I will introduce the subtyping method I use. You're welcome. And this is later I using the ROC to do the validation for the top one by the top 4 panels. So here we show the top 4 panels has relatively high AUC value than the randomly pick up the panels. Yeah. So normally there's 2 groups. The first 4 are for the top 4 panels, and the last 4 are the pick up from the randomly pick up from the samples.
I guess going back to Zhang's point before, what is the purpose of this biomarker?
This purpose just try to find out the biomarker as few as possible for predict the cancer status.
Okay. What is the current approach to? So this is for early detection of cancer, right?
Yeah, because this sample is from the plasma, so its purpose is for the early detection.
Are you competing with mammographies, or are you hoping to identify cancer before something will show up on a mammogram, or?
This is not. It's only for the diagnosis purpose. Diagnosis for the early detection.
I guess what maybe what Zomi was getting at before was that depending on your purpose, you may choose different measurements to optimize, right? If you want a really sensitive test or a very specific test, you may not use AUC to.
Yeah, right. So this is just the general diagnosis purpose. So AUC is the only one I use. But the other, like you mentioned, the sensitivity and specific is only for us to filter out some of the panels, not for the optimization purpose. And this is just now I showed the panels is at the fixed number. That's 5. Yeah. But some people may ask why you choose 5. It's another number is good for combination. So this method is developed for find out what's the best numbers with the highest accuracy. So I use the recursive support vector machine with the cross-validation method. So this method contains much more the accuracy than the previous method, like a support vector machine only support the vector machine with the RFE, the feature recursive feature elimination, and this is our method. So we can see here has much more the higher score than in the testing side than the previous method. So this is the number we find out. So we want to find out the number of the biomarkers with the lowest false discovery rate. So here, the lowest false discovery rate happened at this level, so means 27 biomarker to 15 biomarker. So the minimal number of biomarker is the 15 biomarker. So we use the 15 biomarker as our final panel to predict first train in the training set and then predict for the testing set. So this is the heat map for the testing set prediction. So the bottom are the 15 biomarkers, and this is the green means the health sample. No, green the blue means predicted as health sample. So the first the 40 samples are health sample. So that means like 1, 2, 3, 4, 5. 5 marked as yellow. That means 5 are identified as cancer sample. That's predicted wrongly. The same for the following the 40 cancer samples. That means we have the blue one identified as the blue one. That's wrongly predicted as the healthy sample. As the healthy status, 1, 2, 3, 4, 5, 6, 7, 8. Yeah, 8. 8 out of 40. That means we have the accuracy like 32 over 40. Yeah, that's really like 40, maybe more than 80%. 80% accuracy. So the.
Did you publish this heat map in your paper?
Yeah, I published this one.
Okay. Do you think it's good enough?
You mean 80% accuracy or the?
In cancer research, do you think this has can be useful?
I did a lot of research. I think 80% for the testing side or for the blind data is relatively high enough, because most of the data I analyzed can only have like a 75% for the blind data. But if you build some like a model with some not blind, not with dependent data set, yeah, you can get like a 95% accuracy. That's without any problem. But for blind side, it's really different. I did similar analysis for the AD analysis, Alzheimer's disease analysis. The highest accuracy I can achieve is only 69%. Yeah. So next, I did some the subtyping from the same data set. I used the pathway analysis because already there's some method like subtyping for the molecule, using molecule method, using like genes or proteins. So in this research, I try to use the pathway instead of the genes or proteins. So all the markers shown here are the pathway markers, and the pathways can cluster the 4 subtypes of breast cancer into 2 groups. So one group is lumina A and lumina B. Another group is basal-like and HERB2+. Yeah. This result is consistent with the breast cancer subtyping using like a pathology method. Yeah. The rest I will show the dynamic visualization. So here is the pathway drawn using the cytoscape. So each node size means the total number of proteins in that pathway. So that means across the different subtypes, node size won't change. But what changed is the color. The color means the number of present proteins in that pathways. So here we show the complement and coagulation cascade pathway. So we can see here is relatively red in the lumina A. And when we go to lumina B, there's just a tiny difference. But if we switch to the basal-like HERB2, we can see the red become more dark here. See, become more dark. And when we go to basal-like, it's dark a little bit. So this diagram clearly shows the basal-like clustered together with the HERB2+, and lumen A and lumen B cluster data. So that's we use the dynamic visualization to show the clustering method using the pathway analysis. And later, all the results I did for the data analysis, I use the high performance computing. So the main cluster is located in the Denton campus, which is the Talent 2.0, the cluster. And locally, I built my own server. So within these two servers, they can communicate and transfer the data. And also, I store some of the raw data in the remote server, yeah, and some of the pathway analysis and R code, SASD and Homer stored in my local server. So between these two servers, we can keep communication and do some computation. So that's the structure for the high performance computing. Future plan, because as I mentioned, the pathway analysis, but you probably already noticed, there's no gene ontology analysis. So in the future, I want to migrate the gene ontology into the current iPad. And for my second database, like SSD, which only contains turnip splicing, so I want to add like SNPs and also the post-translational modification. So build a complete protein isoform database and use it for the disease diagnosis and prognosis. The third one is, because this is probably not new now, try to do the alternative splicing, biomarker identification from the RNA sequence. The reason I do this is because I want to do the last one. The last one is for the comparison between the genomic and the proteomics, because some of the paper says their correlation between the genomics and the proteomics is quite low. But if we consider the alternative splicing in the axon-axon junction level, there is some correlation between them. So I want to use this alternative splicing method to try to confirm that or validation that or test that. Yeah, that's the whole purpose. Yeah, that's it. Thank you very much.
Okay, questions? Any questions from you?
How did you select those pathways? Did you filter manually or use some other?
Yeah, you can use like a p-value to filter out some. Yeah. Because you can treat pathway as gene set, right? You can treat the pathway as like a biomarker panel, or just like a single gene or single.
The pathways you select, the genes contained in those pathways may have overlap.
Yeah, they have overlap.
Yeah.
That's right. This is the overlap shown here. The more wider, that means they have more protein or genes share with each other. See, so this pathway has a lot of overlapping, but these two, the overlapping is very small.
But this color, green means down-regulation and the red is up-regulation.
No, it only shows for the pathway we don't have upgrade and down upgrade. We only show the more and the less. The green means the number of protein present in this pathway is very low, probably 0 or maybe 1 or 2. But this the radius means the higher number of gene or protein.
So it's enrichment score.
Yeah.
So that means most of them has low enrichment.
Yeah, right.
No, I have it.
Yes.
So when you put all those pathway database together, like CAG and Reacton, this kind of thing, so you just basically use them as a union?
No, cannot. It's very difficult to merge the pathways from different sources. Yeah. Because they, for example, same pathway, they use a different name, different ID. So I don't know what's the relationship between that the same pathway from different sources.
Question is, if you put all the pathway together, just like a super pathway database, otherwise how can you test this? It's not like you try to curate and annotate it.
Yeah, right. I put all the pathways together, but all the pathways from one source, not from different sources. Yeah, I just use CAG instead. The other like Reacton and NCI curated pathway just ignore. So all pathways shown here is the CAG pathway.
Then if they have any already verified the result in your pathways?
Yeah, like this one. This one was mentioned by a lot of the papers. It's probably important for the breast cancer, for the proteomics.
So related to breast cancer, maybe I ask you one question. There are so many biomarkers reported in breast cancer. My knowledge is very limited, but I didn't see those kind of well-known biomarkers. Have you looked at if you use those known, say, 5 biomarkers, can you have a better performance in your prediction of the outcome, or the heat map in terms of gene expression?
Yeah, there's a quite famous genes or pathways already reported many times by other papers, but in our data set we cannot find that pathways. So I don't know why, very famous one, P53 pathways.
I'm talking about the other slides, the
genes.
Biomarker discovery.
Yeah, same.
So those 15 or the 5, you have several sets of 5 biomarkers. Most of them I'm not very familiar.
Yeah.
But the breast cancer, it's not the best study, one of the best studied cancer types. So have you looked at your results versus those kind of quote true positive biomarkers? Why is this little lower down?
The pathways the markers, the markers we identified is has a small overlapping with current biomarker for breast cancer. Yeah.
Well, what you're looking at the serum biomarkers, right?
Yeah.
So
Not serum, plasma.
So there's not much known about that, right? That's going to have very little to do with the cancer.
Yeah, and
you're most likely going to pick up things that are indicative secondary markers of cancer, like immune response genes activated in the immune system.
yeah, some famous biomarker like BRCA1 or BRCA2 has very low abundance in the proteomics, but we don't know why. They just showed no abundance in the plasma. So.
But on the other hand, you don't care, right? As long as you can find a marker that can give you prediction or whatever power, it doesn't matter what's underneath the map.
I don't know, but at that time there are 4 groups doing this experiment, I think, but only our group published this paper. Other 3 groups has no any publication. I think the reason is try to explain the result is quite difficult. Yeah. So our primary, our original purpose is try to find out those famous biomarker, but just we didn't see it. Yeah.
So what about the original study? Did they publish the biomarkers?
No. Original study, I think one paper published related to the breast cancer is only published in the Journal of Proteomics, and focus on that paper. Focus on not biomarker. Focus on the pipeline and the storage. Yeah. What's the whole pipeline? What's the whole flow chart, data analysis for processing the proteomics, and where they try to storage those data sets.
So your biomarker using proteomic data or microarray gene expression?
Proteomics,
Oh, okay.
plasma proteomics.
So no,
But the method we use can be used for any other type of platform or any other kind of data. But it's just the result is not amazing. Yeah.
any other questions? So maybe ask you one more question. For your secret data, breast cancer, breast cancer because the tissue mainly, to my knowledge, mainly comes from the TCGA data sample tissue repository. So by expectation, if not all, many of those samples, they already published other genomic data sets like
Yeah.
gene expression, methylation. Have you looked at or if you
Yeah.
haven't? what do you think
TCGA
of-
contains a lot of the data for the cancers. For the breast cancer, maybe they have like 110 samples. Yeah. But TCGA doesn't provide raw data. They provide only level 3 data. That means the data already analyzed.
Even so, you can still look at the gene expression, for example.
Yeah, you can look at the biomarker, but cannot use it for the genetic variants. If you try to find sleeves or genetic variants, that data is not enough.
Yeah,
We need the
question
raw
is,
data.
in that table, you have 4 versus 20, those kind of biomarkers. Can you go to the TCGA gene expression data, identify those samples, and see the related gene expression also change?
That,
So that's my question.
yeah, that can we can do that, but we didn't do that.
Okay, if no more questions, then we can finish here. Thanks for the speaker for your talk here.
You're welcome.
Yeah, of course. I'll talk for tonight. Thank you so much for talking to us.
Oh, that's my pleasure. Happy to have that.
Yeah.
We have to big video here, I didn't realize. Yeah, very nice talk, a lot of work. You were, so you were talking.
I'm having lunch with you, right? Jack Chang.
Nice to meet you.
Lots of work, very
You mentioned
productive.
how much KB, so ask him.
Okay.
We'll put it up for the Jeff Chan council. Yeah, the how much KB?
Pharma GKB?
In your one slide, you have how much KB.
Okay.